Okay. Let's circle in. Continue thinking about action potentials. So today we're going to put a cap on our understanding of action potentials and segue into our understanding of what happens when an action potential gets to the end of a neuron to this region that we're going to call a synapse. So we'll move to synapses right now. So at this point, what we know about action potentials, we know that they're different from graded potentials. They're for long-range communication. We send a signal down an axon, and what the signal is made up of is really an electrical pulse. So that electrical pulse is caused by the fact that we have a change to the membrane potential when we have a stimulus, right? So we add up graded potentials. We potentially hit this threshold of -55 millivolts. If we do, a cascade of reactions follow, leading us to this action potential, which is a depolarization, a spike up to +30 in the membrane potential. So we know that this is happening because we have voltage-gated sodium channels that open quickly as their activation gates open in response to this threshold value, allowing sodium to flow rapidly into the cell. We know that we've set up the pressures such that sodium wants really badly to come into cells. Okay. We know that this gets cut off eventually before our membrane potential gets too high, because we have this time delay effect where our inactivation gate on sodium voltage gated channels closes. So now we can't have sodium come in anymore. And at the same time, we see voltage-gated potassium channels opening, meaning that potassium is going to leave the cell more rapidly than it was before, dropping our action potential back down. So dropping us from +30 down to rest, actually even overshooting as we have those potassium channels stay open for a while longer. Okay. So we ended last time thinking about how this creates these things we call refractory periods. So we have an absolute refractory period where we absolutely can't send off a second action potential. And we have a relative refractory period where it's hard to set off a second action potential because potassium is still leaving the cell faster than it otherwise would be. So it kind of washes out the effect of some stimuli. But if you have a big enough stimulus, you might still be able to fire off an action potential during that relative refractory period. Right. So that sets us up to understand a bit about propagation of action potentials. So now we're going to think about, okay, mechanistically, how did we get from the body of our neuron, the soma and dendrites, where it had stimuli coming in, and that little pinching off point, the axon hillock, all the way down the axon to the opposite end where we see our axon terminals and then the synapse. So we're going to think about how this happens in two different types of neurons. So what we're thinking about here is conduction of nerve impulses, so how these charges are traveling, or we can also think of it as propagation of the action potential. You'll see on another side. So how we propagate, so make more of this action potential continuing down the line. Okay. So we're going to have like a wave situation happening. So we're thinking like when you do the wave in a stadium at a sports game. Like when I say a wave, that's kind of what's happening here along our axon. Okay. So we're going to see that we first trigger an action potential in a specific location, so in the axon hillock, right? And then we're going to progressively trigger this section, then we'll trigger a second section, a third section, and so on down the line. Okay. So think of this as like people standing up, then sitting down, but as we move forward, right, once these people have stood up and sat down, these people in this second, a little section are standing up, right? Like, just like we do the wave if we're sitting on bleachers. Okay. So essentially what's happening is that one patch of membrane, so one section of the axon hitting threshold and setting off this cascade, our action potential with respect to positive 30, is the trigger, is the stimulus, is the depolarization to threshold necessary for our next passive membrane to do the same thing. So, one thing that is true about action potentials is that they have a direction to them, right? They're going from where we have our signals coming in and then cell body towards the end of the axon. They don't like bounce back and forth, right? That would be confusing from a signal perspective. That would be really messy information for other neurons to receive. So we can figure out, looking at diagrams of an action potential, how and why we see this traveling. So in this picture, so we have an action potential beginning here in blue. This part in yellow hasn't yet been triggered. As we move on these diagrams, here we have a section in red. Here we have an action potential in blue. Here we have no action potential, hasn't been an action potential yet in yellow. So in this question, what I'm asking you is about that second part, where we have a red part, a blue part, and a yellow part. Where is our action potential going to go next? So we're in this central section. We're letting sodium rush into this blue area. And we want to think about where we are next. Okay. Yeah, so this isn't a trick question. So what we are looking at there is that directionality of the action potential. Okay. Right, so this is stepwise. We have an action potential start here near the body. Then the next place that's going to get triggered is a little farther down the axon. Then the next place that gets triggered further down the axon cell. So this is about that directionality. Okay. So we're going to be doing this patch by patch, section by section, down our neurons. And here we're going to first focus on a neuron that we refer to as an unmyelinated neuron. We're going to see later on that myelin is an extra kind of covering around the outside of an axon, and we'll talk about how that makes a difference later. But for now, we're just thinking about a bare neuron, nothing surrounding it, no other like features that we have to pay attention to. Okay. So when we see action potentials moving down unmyelinated neurons, this is going to be relatively slow compared to our myelinated neurons that we'll talk about next. But what we have happening, all right, is that we are piece by piece triggering every little site along the axon. So here we can see, similarly to that previous picture, if we've got site A in here near the original site of our action potential, from near the axon hillock, which is that pinching off point from the cell body, that gets depolarized. Then progressively, we have the next site depolarized. That depolarization spikes to 30, so that's the action potential. And then on down the line, all moving in that direction here. Every site that gets triggered, we've mentioned before, an action potential is an action potential is an action potential. They're all or nothing. They're all the same. They spike to positive 30, they drop back down. Okay. So this is going to be moving down the line, down the axon, at that same amplitude, that same magnitude, that same strength, the full way. And that's important to mention because that's different than when we talked about graded potentials. Talked about graded potentials decaying as they move from where the stimulus hits. That doesn't happen with an action potential, right? So you can have a really long axon. You're not going to end up with a really weak signal at the end because we're regenerating this the same all the way down the axon. So in those diagrams where we have site A, site B, site C, what's happening is we trigger first site A, then we trigger site B, then we trigger site C. Don't travel back. So once we move to site B and we're triggering the action potential on site B, we don't trigger both neighbors, right? We don't trigger site C further down the axon and also site A. We only trigger site D. We only trigger the one that's farther down the line towards our next neuron. We're going to think about why. So this does relate to the refractory periods that we talked about last time. Okay, so this is why we talked about refractory periods before we talk about how these actions affect. So I need to take a look at these statements about absolute and relative refractory periods and how that links up to what's going on with potassium and sodium. Which of these would be true for site A? It just fired off a spike to positive 30. Okay, cool. We got a little bit of diversity in answers, but we mostly landed on this correct choice. So this is an important impact of refractive periods and how they work, right? So the reason for this directionality, for the fact that we just send that action potential down in one direction, is because the sites closer in towards the cell body are in a state where they physically cannot be activated again. It's just not possible, right? There's no way to re-trigger that patch of membrane that just sent you the signal, right? So this is really important because we want our action potentials to have direction. We're trying to send a clear signal to the next cell that we're talking to. So very useful that this is how absolute refractory periods work. Okay. So we haven't come all the way back down in that first patch of membrane yet. We spiked to positive 30, right? And we're on our way back down, but that means that we've closed up that inactivation gate on the voltage gated sodium channel. It takes some time to reset and reopen. We can't control that time. So there's absolutely nothing we can do to re-trigger site A. So, yeah. Okay. So here's what we see happening, just zoomed in a bit. So we've already talked about the patchiness that we're going patch by patch from the axon hillock down the axon and then towards our axon terminals, which are these little spaghetti fingers at the end. Okay. So our setup at rest is we've got more positive charges outside, lots of sodium. We've got negative charges inside. We know we've set up pressures such that sodium wants to come in, potassium wants to go out. Okay. So that's at rest. And then what we have during our initiation of an action potential, so the first time we see this happening, once we have this initial trigger, here at the axon hillock end, we're going to see that we let a lot of positive charges rush in. So here are our sodiums. This is our positive 30 spike. We got lots of sodium ions come into the cell, right? Fundamentally, that depolarization is the signal. Right? The action potential, if we're looking at all its phases, has an up and then a down, but what's actually going to have the effect for us is this spike part, this big depolarization. Okay. So ions are physical objects. We are in this fluid environment, so these sodium ions come into the cell, but they don't stay stuck there, right? We don't have a catcher's mitt. We're not trapping them. So these sodium ions start to diffuse side to side, right? So some of these sodium ions are moving to the right and the left to neighboring patches of membrane. Okay? So this has the effect of depolarizing neighboring sites, right? So these sodium ions that came into site A and floated over a little bit towards site B are how it got to its own threshold, right? That's how these voltage gated channels that would be on site B perceived that change to a membrane potential. So then we have the same reaction as we did for site A. These sodiums come in, it's positive charge. Now, when those sodium ions spread out side to side, to both site A and site B, see, right? So the sodium ions themselves don't know anything about the direction, but we only trigger site C because at site A, we're in that absolute refractory period, right? So we've got full sodium coming in that might affect membrane potential, except for the fact that our voltage gated channels are inactive, right? And our potassium channels are open. So there's no re-triggering of the action potential in that location that hasn't fully reset yet. So that's how we move down our axon. Okay. That's what happens in an unmyelinated neuron. What happens in a myelinated neuron is similar, but we've got some changes that make this work a little faster. So myelin speeds up propagation of action potentials. Okay. This is an insulator. So we're thinking about like, you can have a bare electrical wire or you can have an insulated wire. They work differently. Same thing with neurons. So this is our insulated wire. So here we can see that insulation in the form of myelin. So here we have a chunk of myelin. Here we have a chunk of myelin. So it's wrapped in sections around our neuron, right? So on down the axon. Now these sections have gaps between them. So these gaps between neighboring regions that are insulated by myelin are called our nodes of Ranvier. So these are our nodes. Okay. So the reason that this process ends up faster, and the reason we propagate an action potential faster in a myelinated neuron is that we don't have to trigger every single patch of our neuron anymore. We see this referred to as like saltatory, so jumping conduction, where we just trigger these empty spaces, right? So we just trigger this node of Ranvier, then this node, then this node. So we kind of skip some steps, basically, when we're looking at myelinated neurons, which is why these are going to move faster. So when we think about where we would see our different types of channels for ions to move through, we're going to see the voltage gated channels are concentrated in these nodes of Ranvier because those are the parts where we're trying to change voltage, right? Those are the parts where we're actually trying to create an action potential in a myelinated neuron. So there's no point of having a bunch of voltage gated channels here, right? In the middle of a bit of myelin, because it's blocked by this insulation. It's blocked by the myelin. So you wouldn't have ions moving in and out in that region. Otherwise, it works the same way. So in a myelinated neuron, other than the fact that we're skipping some patches of membrane because they're surrounded by this insulating myelin, we still have directionality. So we're still going from the axon hillock down the axon towards the axon terminals. We're still triggering a patch of membrane, having those ions spread out, and then triggering another patch of membrane. It's just, we've got some gaps. We're skipping some sections. We can move a little faster through this jumping action. Okay. So one way that we can speed up conduction of an action potential, say you're thinking about a neuron, like a nerve cell, right, that's going from your brain to your big toe. You don't want to wait forever for that, right? You want to get there relatively fast, but you've got a big distance to cover, right? So one thing we can do is cover the myelin, right? So myelin's going to speed up conduction. The other thing that can speed up action potentials, the thickness of the axon itself. So like the size of the neuron. Okay. So what we're going to see is that bigger neurons, so wider diameter, ends up speeding up our conduction. So in the future, we'll think a lot about flow of like blood through blood vessels and tubes, and we'll think about flow of air through airways. And it's kind of analogous to what we see happening a little bit here in our neurons. So if you want to think of a way that you can remember this increase in diameter speeding stuff up, think about what it's like to drink something through like a really wide milkshake straw, right? You get liquid, lots of liquid fast, versus what it's like if you try to drink through like a coffee straw or a cocktail straw, right? Like really hard to get a bunch of liquid into your mouth. That's analogous to what's happening here as we see our ions moving and spreading this charge. This increased diameter reduces our resistance to general spread, general flow of things within a tube. So this basically speeds up how fast this charge is traveling, the way we'll see big diameters speed up how fast fluids travel and how fast air can travel. Okay. So the fastest neurons, the fastest action potentials, are going to be where we have a big neuron with myelination on it. So we can see, for example, we have a big neuron here. It's going fast, right? Way more meters per second than our little tiny ones here, right? So here we have a little tiny diameter neuron going very few meters per second. So this is part of why when you look in lab, you're going to see these larger nerves going towards our like farther out body parts than when you look at like little tiny cranial nerves in the brain just going, you know, to a little muscle by the eye, right? So if we think about those sizes that we're going to see and that we see in the body lab, this kind of lines up big. Okay. So we're going to move forward and think about synapses. When we think about synapses, what we're talking about is once we get an action potential to the end of the axon, to its axon terminal, what does that do for us? Like, how does that actually help us communicate between cells? That's where our synapses come in. So we're going to find that we have a couple different types of synapses. We're going to have chemical synapses and electrical synapses. We're going to think about the role of neurotransmitters in our synapses, how they're a response to action potentials, and then what they do to the cell that's receiving this signal. So we're going to start by defining a synapse. So when we talk about a synapse, all we're talking about, I'm drawing because it's a lot of text, right? So all we're talking about is we have one neuron terminal talking to another cell. All right. So this area in the center where we're communicating is our synapse. Okay. This is a functional connection between two cells that are talking. In the CNS, which is our central nervous system, so we're thinking brain, we're thinking spinal cord for central nervous system. When we look at these synapses, it's going to be one neuron, so one link nerve talking to another nerve cell. So one neuron talking to the second neuron. When we look at our peripheral nervous system, which are the nerves going out to like your fingers and toes, the rest of your body, and even the brain, spinal cord, we're going to see that we have our neurons talking to other types of tissue. So we'll see later on that sometimes this second cell will be like a muscle or a gland or something. But functionally, we're going to have this same sort of connection where we have a cell that's sending out a signal and a cell that's receiving a signal. Okay. So when we talk about this signaling, we're going to be focusing on our central nervous system for now. So we'll start with this first connection type, this neuron to neuron communication in the central nervous system. So we're thinking like the brain, right? Where we are right now. So when we have two neurons in a line, we can use our abbreviations for before and after, right? So we have a pre and a post, right? Synaptic neuron, right? So presynaptic, before the synapse. Postsynaptic, after the synapse. So we don't have to number them. Neuron 1, neuron 2. We've got our presynaptic neuron and our postsynaptic neuron here. Yeah. So our presynaptic neuron is going to act kind of as a stimulus for our postsynaptic neuron to react to things. Right, so that presynaptic neuron, that first one in the line, might be signaling onto the cell body as I've drawn it here. We know that sometimes neurons have these spikes looking kind of like halo or lines made or something on them. They have dendrites, so specialized areas for receiving signals. So presynaptic neuron could go to a dendrite. Sometimes we'll see presynaptic neurons actually synapsing, so creating this kind of connection onto the axon of the second neuron, onto the axon of the postsynaptic neuron. So that's possible too. Any of these connections, we would just describe based on which two parts are touching, right? So here, in this example, I've probably drawn an axosomatic connection, right? So from the axon or axon terminal, right, this little button onto the cell body, the soma of our postsynaptic neuron. I didn't draw any dendrites on this one, which is why it's not so dendritic. But if you had that presynaptic neuron and its axon terminal talking to a dendrite, axodendritic synapse. If you had that axon terminal of the presynaptic neuron synapsing like somewhere down here, right, that came in kind of from the side, that would be an axoaxon synapse. Most synapses are axodendritic. Most of them just have communication in one direction, so from presynaptic neuron to postsynaptic neuron. That's like what a synapse is, this functional connection. I think that'll get a little more clarified as we think about what our electrical and chemical synapses is. So what I mean there by functional connection, get a little clarified. Being a little vague because these two types of synapses are pretty different. They do work pretty differently. So exactly how to define this functional connection can be a little tricky. Okay. So in this image, I'll pull them out separately, but what we're looking at are two different types of neurons. So we're looking up top at a synapse where we have these gap junctions going all the way through the membrane of one cell into the membrane of the other cell, connecting cytoplasm to cytoplasm. Right, so along this connection, we could literally have ions move from the cytoplasm of a presynaptic neuron to the cytoplasm of a postsynaptic neuron. So we're going to have electrical synapses. And in the second one, we have two neurons that aren't physically connected, right? Like we don't have a tube running from one to the other. We have this little space, right? But what we have happening is our presynaptic neuron releasing a chemical that is then received by our postsynaptic neuron. So that sets us up for these two types of synapses. The first one is an electrical synapse. When we're thinking electricity in cells, what we're thinking about, action potentials, what we're thinking about is movement of ions, changing membrane potentials. Fundamentally, from our cell perspective, that's really what electricity is. That's why we're measuring those changes to the membrane potential in millivolts. Okay, so this electrical synapse with these gap junctions forming a tube that goes all the way from the cytoplasm of one cell to another cell, we can have really rapid communication as things move from cytoplasm to cytoplasm with very little barrier, right? They don't have to do much of anything other than encounter this gap junction, right? If they come next to this gap junction, it's just a tube that goes to the next cell, they can click on through. Okay. So we have some electrical synapses like this that go neuron to neuron. We have some connections like this that go between neurons and glial cells. Glial cells we'll talk about later, but think of them like protective cells in your brain. So other types of cells that you have in the nervous system that aren't directly firing action. So easiest thing to think about as what might be going through these gap junctions in the electrical synapses would be ions. We could have other second messengers, so other kind of messaging molecules produced inside these cells traveling through. Because we have this just kind of uncontrolled tube between the two cells, that's what the gap junction basically is. We don't have a lot of regulation on that. This is a case where you might have signals go kind of back and forth. Okay, so usually when we think about a synapse, we're going to be thinking about presynaptic to postsynaptic. This would be the exception because we can have something travel this way through a gap junction. We could have something travel that way through a gap junction, right? So this does make bidirectional communication possible. So at this point, at this synapse, in terms of reactions that we might have to this communication, we might have excitation. Remember, excitation is when we're more likely to depolarize, so we're making the charge a little more positive. Okay. At this same synapse, we also have the possibility for inhibition. So inhibition is like suppressing action. So this would be anything that's making our membrane potential lower than rest, so lower than -70, gonna make it difficult to fire off an action potential. In terms of places that you'll see these electrical synapses, so you see them in the retina of your eye, okay? You see them in the cortex in your brain as well as in the brain stem, so controlling breathing in the brain stem especially. You also see them in the hypothalamus, which is going to be really related to how we release hormones. We'll talk in detail about that in our endocrine system. So you see these electrical synapses in places where you need really rapid communication, right? Like looking at stuff and understanding images with your retina, with the eye, requires really fast communication because your eyes can move pretty fast. Things can move across your field of vision pretty fast. These other places also want really rapid kind of communication. That is all we will be saying about electrical synapses. Just that one thing, we've got some places, they're different than chemical synapses, but that's about it. When we talk about synapses moving forward, we're thinking about chemical synapses. So chemical synapses you're going to see everywhere else. Chemical synapses are when we're going to be talking about neurotransmitters. Chemical synapses are where we're going to get really in detail in terms of what part of which neuron is touching our postsynaptic neuron and how they're all kind of working together. So this is usually where we're focusing on labeling all these different neurons. So presynaptic neuron is going to be before a synapse, right? So before it connects to a postsynaptic neuron. Right? But as you will see, we're going to have to focus in and specify where we're actually talking about, because neurons are talking to a lot of different places all at once. Right? So in this example, you can see that we actually have multiple presynaptic neurons here. This postsynaptic neuron is getting signals and messages coming from, in this image, 3 different neurons. So that would be 3 separate synapses. And so we'd be focusing on the synapse. Right, so then we have presynaptic 1 talking to our postsynaptic neuron. And here we have a different presynaptic neuron, so we could still, again, label 1 to another presynaptic neuron on that synapse talking to our postsynaptic. So eventually we'll get to how does the cell handle potentially conflicting information coming from different places. Okay. But for now, we're going to be focusing in just on one synapse and a given synapse, thinking about a presynaptic neuron, talking to a postsynaptic neuron, across a synaptic cleft. So that gap we see, a tiny little gap here, that I've also been drawing in my images, so the little blank space, that's our synaptic cleft. It's like a canyon between the 2 cells. Pretty small, 30 to 50 nanometers wide, but it is there. There's a space. We're not going cytoplasm to cytoplasm. In our chemical synapses, we're going to have unidirectional communication. So we're going to be going from the presynaptic neuron to the postsynaptic neuron, and we're thinking about where's the signal. Usually from the axon terminal of the presynaptic neuron to probably a dendrite on the postsynaptic neuron, but there's lots of variation possible. We really won't talk about dendrodendritic synapses, but those are even additional 4th kind of possibility beyond the ones that we're going to be focusing on. Okay. So we're about to talk about the steps of communication at a synapse, which is going to focus in on sending out neurotransmitters and receiving them. So when we talk about cells talking using neurotransmitters, what's actually happening from a mechanism perspective is that we have a neurotransmitter that's acting as a stimulus for a graded potential in a cell that's receiving that signal. So using the terminology that we've just kind of learned, how might we refer to the cell that was making that neurotransmitter? Okay, awesome. Yeah. So the cell that's going to be sending out the neurotransmitter is our presynaptic neuron. The cell that's going to be receiving our neurotransmitter is going to be our postsynaptic neuron. So when we're looking at diagrams that look like this, pay attention to the labeling on the cells, right? Because that's going to tell us where we have our presynaptic neuron, right? So first thing we want to know is we've got cell 1, our presynaptic neuron, and we've got cell 2, we've got our postsynaptic neuron, right? So we're zooming in on the synaptic cleft right here, just tiny little area where we have this communication synapse between our presynaptic neurons. So other things that we can see in this synapse before we get to our specific steps are some features that are going to be associated with the release of neurotransmitters and with receiving neurotransmitters. Okay. So one notable thing that we see in the presynaptic neuron, we got these pockets. Okay. So these pockets are called synaptic vesicles. Okay. So synaptic because we're at the synapse, right? That's our location. Vesicles, because a vesicle is basically the word in biology for a little baggie of stuff inside a cell. So we've got a membrane here, right? That's what this circle is made of. A membrane surrounding the contents of the vesicle. So inside these synaptic vesicles is where we have our neurotransmitters stored. Right? So our neurotransmitters are going to be the molecules that we see in these synaptic vesicles. Other things that we can see on this presynaptic neuron. Got our neurotransmitters ready to go. Another thing we're going to notice, we've got a new type of voltage-gated channel. So here we see a voltage-gated calcium channel. This is adding on a third type. We've had voltage-gated sodium channels. We've had voltage-gated potassium channels. Now we have voltage-gated calcium channels. So these voltage-gated calcium channels are specifically going to be located in the axon terminal of that presynaptic neuron. Okay? That's where we see these channels. The other thing I'm going to jump to, since we're going to focus on presynaptic neuron, just all its pieces, is this reuptake molecule. Okay, so the other thing that we can see here is that we have an additional membrane protein labeled a reuptake molecule. We're going to see that that's involved with what happens after we release our neurotransmitters. So then looking at the postsynaptic neuron, so the second one, I want you to notice right off the bat, there are no channels here on this postsynaptic neuron. Okay? We don't see any channels yet. All right, what we see is a receptor. Okay, so we got a receptor. So this is going to receive signals, hence the word receptor. And we see an enzyme. So we see an enzyme that can break stuff down. Those are the important pieces of cellular machinery that we want to be aware of as we start to piece together what happens with this synapse. So here's the whole big flow that we want to understand and be able to put in order for ourselves. So here's the story of how our synapses are communicating. And we already know that in our presynaptic neuron, we had stimuli, they built up a charge that took our neuron to threshold, causing an action potential, spiked up to positive 30. And we did that patch by patch all the way down our axon till we reach the axon terminal. So where we start with synaptic communication is that action potential getting to this location, right? So the first thing that has to happen for us to communicate with our postsynaptic neuron is we need an action potential to reach the end of our presynaptic. So an action potential is fundamentally this depolarization to positive 30, right? So we sent a wave of electrical charge. The electrical charge is now here in the axon. That's why we have these voltage gated units. So here we see what that voltage gated calcium channel does in response to this change to the membrane. So as we have this spike to positive 30 coming in with our action potential, that's the trigger to open these voltage gated calcium channels. We don't have to worry about different types of gates with these ones either. That's just for our sodium channel. So we're going to think about the calcium channel as just opening when we have our action potential shot. So the electrochemical driving force on calcium is such calcium wants to come into cells, so just like sodium. So calcium wants to come into cells, sodium wants to come into cells. So far, we just got potassium that wants to leave cells, where we're focusing on the 3 cations that we mentioned in this unit. Okay, so calcium comes in. So now we have the axon terminal that's full of calcium that wasn't there before. That calcium is the trigger for the next step. Once we have calcium here in the axon terminal, that's our trigger for these synaptic vesicles to do something. Specifically, the something is that our goal is to release our neurotransmitters that have been stored in these vesicles, stored in these pockets. All right, so here we can see these vesicles docking, right? So basically attaching to the membrane here next to the synaptic cleft, and then secreting, so sending out the neurotransmitters that were inside. Okay. So the docking and secretion happens next. This is basically exocytosis, if you remember that term from before when I mentioned it once, or more likely from your previous classes. Okay. So once we have that neurotransmitter in our synaptic cleft, it's got to diffuse across a little bit of space, and it's got to run into a receptor. So here we can see our neurotransmitter, this little triangle, bound into the receptor. So the receptor is some type of protein, right? So this is a membrane protein. So when we have this receptor bind our neurotransmitter, it's going to change shape. Right? Proteins are all about shape. Their shapes are really dependent on their chemical properties. When you touch them with something new, they change shape. We talked about that before when we talked about enzymes and how that works to lower activation energy and control reaction. Receptors aren't doing a chemical reaction between 2 molecules, but they still have this shape change in response to contact with a signaling molecule. So when we have this receptor come into contact with our neurotransmitter, the neurotransmitter itself doesn't go into the cell. Right? That's why I wanted to point out there are no channels on this diagram yet. What happens is that receptor changes shape. And as it changes shape, then we're going to have new things going on in the cell. So our cell is going to have some type of response. And then our signal is gotten all the way to our postsynaptic cell. So the rest of this is just about how we get rid of the neurotransmitter, right? So some neurotransmitters get broken down by enzymes, and then those chunks just kind of diffuse out of the cleft. Some neurotransmitters get pulled back into our presynaptic cells. So we're basically recycling using this field. Okay. So we'll stop there today. Thank you all. And next time, we'll talk about some different receptors and some different reactions that they have.